रेगुलर नॉट एक्सपेरिमेंट चला पेपर से फिनिश ना पेपर देखना पेपर से फिर मैं आपको जरूर में काम करना ये चश्मा लगाना चश्मा है ये कुछ किया भी क्या Oh my God, zero. मैं बोली तो पता नहीं कितना साल हो गए सुनना पड़ता है तो मिला एक मिला था एक दो ये मतलब मैं बोल मत किसी को कितना मिले देखो हम लोग आ रहे मुझे अभी 14 मिला था उसके लिए मुझे मारी हुई थी एमसीक्यू इतना इजी एमसीक्यू है ना हाँ बैठो आप पहले पढ़ो हमारे साथ वापस अच्छा कोई एक मार्क्स वगैरह मिनिमम 5 9 इसके मैक्स उसमें भी वैसे बोल रहे हैं वैसे लिखते हैं यहाँ पे बैठे अपने पीछे बैठे हैं कब से तो उठ रहे हैं बोल रहे पागल है एक दिख रहा है पागल सब अंदर कुछ है उधर पोस्टर सच में अब आप आएंगे क्या हुआ कौन अबे यहाँ से दिख रहा है अच्छे से देखी तो क्या तेरा शक्ल देखना लोग तेरे शक्ल के लिए इतना ये कर कौन फेमस है ऐसे हम सबको खड़े लोग को नहीं रेस्पेक्ट रिमोट नहीं है नहीं मिला तुम लोगों को सर
ऐसे कर लो
इसरो रिमोट क्या दिख रही है भाई ये शादी का ये हाँ आइए क्या कर रहे हम पेपर्स ये पेपर्स का क्या लग रहा है इतने कॉमर्स है सब ब्रांच में आइए अच्छा पेपर देखे देखे तुम लोगों ने ना ऐसे इतने शरीफ मत बनाओ इतने शरीफ मत बनाओ मैंने देखा
मैंने वहां संस्करण देखी
मैं थोड़ा सा जैसे मैम को थोड़ा क्लियर करूँ ना क्या है वहाँ पे कुछ हेल्प हो जाता है क्या आपको स्कूल लेवल में बहुत जाता है ये अभी जा रही है नहीं जाता हम लोग एक इसमें हम लोग एक के लिए लोग 2-3 वही तो मैं बोल रही हूँ सब लोग मैं पहले जाती थी पर नंगा वो क्या करती है ना 4 सब्जेक्ट एक ही मैम लेती है ये जस्सी बोलकर है ना शी वाज अ वाइस प्रिंसिपल थी वो तो रिटायर होकर अपना
अपना खुद एक ऐसे नहीं मतलब लाइक को उधर से आगा हुआ था हाँ वो गई हाँ ठीक है जो भी है पर she has connection with जो साधु वासवानी लोग हैं ना उनसे पूरा कनेक्शन है तो फिर
उसको वो मार्क्स वगैरह अच्छे से मिल जाते हैं
और प्लस वो वेस्टर्न स्कूल में तो बोल समझ से कि बोली का ये अच्छा मतलब पर ये प्रॉब्लम है तो मैं 4 सब्जेक्ट्स एक दिन आप टीच करती हूँ मैं पूरा प्रेशर आता है और उसके ऊपर से मैं अकेली हूँ ये लोग साधु वासवानी निकाय में तो मैं अकेली रहती हूँ जो उसमें मेरा पोर्शन नहीं कंप्लीट होता है तो वो पोर्शन होता है और नहीं होता है सच्चे में तो कुछ नहीं पता
दया बहुत शर्माता है ना
जरिया
पहले सिखाती थी
बहुत अजीब है वो इंसान ये पूरा ये तो है कि वो बहुत आउट ऑफ द बॉक्स सिखाती है आउट ऑफ द बॉक्स पढ़ाती है लेकिन अगर मैं दूसरा स्कूल आएगा तो हमारे को फोकस नहीं जिसका मेजॉरिटी है उसका देगा बस
अच्छा और कौन सा जा तुम दोनों तो कभी गए और तुम दोनों
जाने की सोच
तो
तू गया था ना आपके पास
ठीक है आपको यहाँ पे समझ में आ रहा है
हाँ वो तो है
टेस्ट है तो मुझे बताओ वैसे मैं अदरवाइज में जो भी है क्वेश्चन पे टेस्ट करवाऊंगी मर्ज होके वैसे जंबल हो कैसे सीन कर रहे हैं
आता ते यूट्यूब वर ना त्याचा पोर्शन आला म्हणजे सगळा नाही पण काही काही आला
आला है अच्छा जो दिला था ते मर्ज होऊन चैप्टर्स होते ना तो ये मार्क्स भी ऑटोमेटिक बढ़ते होंगे ना उनके स्कूल में बच्चों के अच्छे चलेंगे चलो ऑल द बेस्ट
Good morning. How are you finding the morning lectures here? Yes.
Sleeping, dozing.
no
No, actually, I'll take a nap during 15 minutes break so that I can get refreshed for the next lecture.
During these breaks?
Yes.
Wow, good. Nap she's taking. Take enough, huh? You make a habit because already, but I think all of your schools are morning schools, no?
Yes, ma'am.
7 o'clock schools?
Yes, ma'am. 5 o'clock. Extra. I need to study to start the previous time.
So all of you are used to it already. But sitting continuously for 6 hours, which you generally don't do in school, no? School में first period में concentration रहता है, second period में थोड़ा कम, third period में और कम, है ना? So now here you'll have to be attentive. 6 hours meaning we are really teaching the 6 hours. So by the end of May, you will see quite a lot of portions are already over. Who is absent today?
7 days
7. How many of them are there? 5 of them. Why? They all have exams now. Acha, they are having exams now.
Yes, ma'am. But Arya is also not coming because he has school till 15. After that, he'll come.
Acha, they all have schools. Which school is still on? 7. Old Crest. Only one of them. One more from Old Crest.
And one from Suda.
You are the only one from Pune. Now let us continue. We have.
my main students are absent
I have to.
7 days and the one was from both the seats also absent
So
From 10
Aradhya.
this topics again has to be repeated. That is what I'm thinking. Now we are talking about genetics. No, we had already started with the concept of genetics, and we already studied some few definitions. We had already started some few definitions. What did we learn? We had learned some few concepts, that is dominant. What is a dominant allele?
Dominant allele is the allele which expresses itself without consideration of the recessive allele. For example, a
No,
black.
you have to write a specific definition. Define dominant allele. What are you supposed to write? An allele which expresses homozygous and heterozygous condition. Or you say the other way, an allele which can express in presence of another contrasting allele. An allele which can express in presence of a contrasting allele, that is always a dominant allele.
What do you understand by heterozygous? We had studied what is the meaning of a heterozygous condition. Heterozygous condition would mean that in a pair of chromosome, the gene has two different dissimilar alleles for that particular gene. So when in a pair of chromosome there are two dissimilar alleles for a gene, it is a heterozygous condition. And if it is two similar alleles, then we say it's a homozygous condition. So this pair of terms should be very, very distinct. If I give you, if I ask you, give me an example of phenotype, then what can you tell me? One example of phenotype.
The visual expression.
That's a definition. Visual expression of gene. That means visual expression of gene. Can you give me one example? Phenotype.
For example,
Black color of the eye.
curly hair,
And the pair of allele that is responsible for the black color. What will you call this as? The pair of allele which is responsible for the black color, the pair of allele which is responsible for the curly hair. What is this called? Genotype. Genotype. So all these terms should be very clear. Genotype, phenotype, homozygous, heterozygous. Then we had talked about dominant, recessive. Then we had talked about gene and allele. What was the definition of alleles? Different forms of genes, alternative forms of genes. And so also what are traits?
different
Forms of our character. Okay. So all these definitions that we had studied the other day, let us move on with another concept. Now, variations, we'll talk about variations little bit. Before that, we were talking about chromosomes. No, we were talking about homologous chromosomes. Now, here, when I talk about chromosomes, let us start with this topic. How did we define the word chromosomes? Chromosomes are extensively
highly constructed.
chromatin. Extensively condensed chromatin threads. Highly condensed chromatin threads. And where do we find chromosomes? Always in a cell which is ready for the process of cell division. And now we talk about interface chromatin. Interface, if you talk about an interface cell, then it has to be chromatin. We don't see chromosomes there. And now when we are talking about the concept of chromosomes, something more that we come across is that these chromosomes, how many types, in what respective number they are there in human cells. So if I talk about that the two types of chromosomes, what are the two types of chromosomes that we have? We have one type of chromosome known as autosomes, and this autosome is actually the first to the 22nd pair. The first to the 22nd pair of chromosome that we have in our body are the autosomes. What is the basic function of the autosomes, or what type of genes do these autosomes carry? These autosomes are actually known as also somatic chromosomes. So when we are talking about autosomes, we also understand autosomes means somatic chromosomes. So they would carry genes. These chromosomes would carry a whole lot of genes which would regulate the somatic characteristics of our body. What is the meaning of somatic characteristics? Characteristics, something like associated with the structure, function, development of any type of a organ tissue. So if I would understand that, what would the liver cells be like? What would be the shape of the liver cells? What should the liver cells produce? How many pancreatic cells should be releasing enzymes and how many releasing hormones? So if I have to understand that all the structural and the functional aspect of the somatic organs of the body, let it be the muscle cells and the bone cells and the nerve cells, etc., etc., if that is what we are talking about, that is the role of the somatic chromosomes or the autosomes. So how many pairs of autosomes do we have? 22 pairs, a total of 44. So a total of 44 chromosomes in every cell. Irrespective of testes, ovary or any other cell of the body, we have 44 chromosomes, 44 autosomes. 44 autosomes. And next is we are talking about the allosomes. Now that's the second category. We are talking about allosomes. This allosome is the 23rd pair. This allosome that we are talking about is the 23rd pair, and these chromosomes are also known as the sex chromosomes, because these pair of chromosomes, if you see, carry a whole lot of genes, and these genes will only and only regulate the reproductive functions in our body. Something like what type of enzymes and hormones associated with reproduction, the entire process of development, maturation of the reproductive glands and structures, and the accessory organs, if I have to talk about the coming up of the beard and the scanty body hair and the voice and etc., which are all associated with the reproductive features. If this is what has to be regulated, then these set of genes, these set of chromosomes will regulate all these functions. So how many pair I am talking about? I am talking about one single pair of allosomes. But these allosomes, if I talk about the male and the female, is there any difference? Yes, there is. When I talk about the males, this 23rd pair in the case of males would be something like this, X and Y. And in case of female, it would be something like this. So here in case of males, we will come across that the males, that's why when it comes to the concept of cell division and gamete formation. Now, what is gametes? Specialized reproductive cells that the male and the female body can produce. So when it comes to the concept of gamete formation, I would understand that the males, that's why, are known as heterogametic. What is the meaning of heterogametic? When it talks about the rest, autosomes are all the same. But if I talk about a male cell, a cell of the testis resulting in the formation of gametes, there can be two types of gametes that the male cell can produce. How? If I talk about a cell of the testis, let me just talk about a cell of the testis, and this cell of the testis will have 44 autosomes, and how many allosome? It will be 2 allosomes, and what are those? X and Y. So this cell, when it undergoes the process of gamete formation, first of all, there will be 4 gametes formed, and the concept of meiosis already you know. Now this gametes, how many chromosomes, how many autosomes and how many allosomes? I would write that there is 22 autosome and X, 22 autosomes and Y, 22 autosome and X, and 22 autosomes or Y. That means I'm saying that these males, they can produce 2 types of gametes. Half of the gametes carrying X and half of the gametes carrying Y, because this pair of chromosomes will get split up. The 23rd pair, when it gets split up, half of the gametes will get X chromosome, half of the gametes will get Y chromosome. So are we not talking about different types of gametes that the male body can produce? Yes, hence the word heterogametic. Heterogametic. But if I have to talk about the female, here we will say the females, we are homogametic. That is, if I have to talk about same concept like this, and if I'm understanding, let this be a cell of the ovary, and how many autosomes? 44 autosomes. How many pair of sex chromosomes? 1 pair. And if this has to undergo the process of gamete formation, obviously we are talking about that 4 cells. If it is meiosis, it should be 4 cells. But now what would be the division? 22 autosomes and X, 22 autosomes and X, 22 autosomes and X, and 22 autosomes and X. So all of them are almost the same type, are actually the same type. Because this X, a pair of X chromosome gets distributed, and the set of genes and the pair of genes in these X chromosomes are more or less the same. So we are saying homogametic. Okay, homogametic. So this, first of all, to understand that how many pairs of chromosomes that we generally complete, or how many pairs of chromosomes are basically present in the gametes. Now, when I talk about these gametes, these are specialized reproductive cells. Are they able to multiply on their own? No, they don't. They are not able to multiply on their own. Why? Because they have got only half set of the chromosome, one half set of the chromosome. What is this one half set of the chromosome known as? They have haploid number of chromosomes, remember. They have haploid number of chromosomes. What is the meaning of haploid? They have got just one half set. What is the meaning of one half set? How many chromosomes in each?
20.
How many chromosomes in each? 11. How many chromosomes in each? 23. And our total is 46. So they have got just half of it now. So with this half chromosomes, that's the reason each of these cells, they cannot undergo the process of division and growth on their own. So these gametes, that's why they have a specialized lifespan also to talk about. Each of these cells, if I talk about the male gamete, it has got a lifespan of just 2 days. And if I talk about the female gamete, even lesser, lifespan of just a day. So within this period, if the male and the female gamete can undergo the process of fertilization, then the diploid number is set back again. Then the zygote is again having the 46 number. So a cell which is having 46 chromosome now it can undergo the process of growth and division on its own. So that's the reason we say that if it is a skin cell, can it show the process of growth division on its own? Yes, because the skin cells and the muscle cells and the bone cells and the liver cells, they are all undergoing the process of mitotic division, and every time the number of chromosomes are 46. Exceptions are the cells of the reproductive organs, the gametes, which have just half of the number of chromosomes. So that is the reason most of the time in that separate chapter also we will come across, and most of the time these gametes are formed and they die out because fertilization would not happen every time. So what will happen? I mean, how will these gametes continue again their process of formation and growth? Only when the process of fertilization can happen, and that also fertilization has to happen within that short period of lifespan. Within that short period of lifespan. I already asked you, you have to go through that table of characters and traits. Last time I must have told you already. Sometimes you are asked also questions on that. Now, next is when we talk about the next concept, about the concept of sex chromosomes and autosomes, we had already talked about. Let us now look into the concept of sex determination. Yes.
दोनों बंद किया मैंने मतलब इसमें
Okay, next is we look into a concept of sex determination. Now let us try writing the concept of sex determination properly. How do we write? I want all of you to write it down. The concept of sex determination. You will show only 2 types, no need of showing all the 4, because types only 2. When you are asked to draw the flow chart for the process of sex determination, this is what you are supposed to draw with little bit of an explanation. What do you explain here? You are supposed to write down that these are the cells of the testes and the ovaries, so these are the diploid cells, 46 chromosomes. But now we are talking about the types of chromosomes also, not only the number. We are talking about 2 types of chromosomes. These are the cells of the testis, ovary. Number would be same, type would be exactly same. Where is the difference? The difference is only in the 23rd pair. How many chromosomes does the gametes have? Again, take a note always. The number of chromosomes in the gametes always 23, not 23 pairs. Sometimes when certain questions come, many students end up writing 23 pairs. It is just 23. And this is what we talk about as the haploid number. And by the process of fertilization, again, the restoration of the chromosome happens. What is the meaning of restoration? Again, coming back of the 46 with the 2 types, autosomes and sex chromosomes, exactly. So this is what we are talking about as the concept of sex determination. Now, when we talk about certain questions on the concept of sex determination, the sex of a offspring, is it because of the mother? Is the mother responsible for the sex of the offspring? No, not at all. Is the father responsible for the sex of the offspring? Not at all. When we are talking about the father produces a pair of chromosomes X and Y, but the sex of the child, neither the mother nor the father is responsible, because fertilization is just a matter of chance. So when you are supposed to answer questions, say that you are asked sometimes questions. Is the father responsible for the 3 consecutive daughter child formed or given rise by a couple. A couple gives rise to 4 daughters in a row. What is the probability that the couple would give rise to a son in the 5th pregnancy? 50%, because the male will produce 2 types of gametes. But which gamete will fertilize the ovum? That is just a matter of chance. The process of fertilization is neither regulated by the female nor the male parent. So actually the male parent is also not considered responsible for the formation of a male or a female child, even though we generally understand that the males can produce 2 types of gametes, but are we to make them responsible that it is they who can decide the process of fertilization? No, they cannot decide the process of fertilization. Fertilization is just a matter of chance. Just one concept to now just discuss on. We are going to talk about this in detail in the later part, I think, when we talk about the laws. What is variation? Now, just a small definition as of now. When we are talking about the concept of variation, did we talk about little when we were talking about cell division? Did we introduce this concept, variation there? Now, what is variation? Variation means, now that we are talking about the whole lot of gamete formation and coming together of the chromosomes, no? And remember when we were talking about meiosis, there I had also explained you one concept of crossing over. Do you remember that? During the process of meiosis, I had explained you one concept of crossing over. What happens there? What happens there? Who remembers? What was homologous chromosome I told you? This chromosome from the father and this chromosome from the mother. Say suppose they will always align side by side. And why do they align side by side? Because of their sequence of genes, which would be very, very similar, because we are talking about a species. So now the mother and the father's chromosome, the paternal and the maternal chromosome coming together and forming a pair, and this pair is known as the homologous pair. This pair of chromosome is known as a homologous pair. So actually every pair of chromosome that we have is a homologous pair. Every chromosome that we talk about, you'll have to put this off. I'm having a bad cough. You can put that on. Where's the remote?
That's off only.
That's off only?
Yes, sir.
Sure, no?
Yes, sir.
Now, when we are talking about the homologous pair, remember what did we talk about? Every pair of chromosome that we have in our cells is a homologous pair, because every pair has got one chromosome from the father, one chromosome from the mother. No? But something to talk about with relation to variation. Now what? This pair of chromosome, it will undergo the process of duplication, something like this, and it will undergo the process of duplication, something like this. So if this process of duplication is true for both the chromosomes, we also come across that these 2 newly formed copies, they always have a tendency of overlapping. And now after overlapping, when they turn, twist and they will separate, we are going to come across that this part of the chromosome will get attached with this, and this part of the chromosome will get attached with this. So what is actually happening? It is we talk about a process known as crossing over and formation of recombinants. So I may draw something like this, that if this is a chromosome getting formed after the process of crossing over, something like this may have happened, where perhaps we will come across that this will have certain parts of the chromosome like this, and where this will have certain parts of the chromosome this way. So if this is what is happening, this pair of chromosomes were not like this in the parents, but it has happened in our cells. Then what is this called? This is what is called recombinations formed. Now recombination meaning the sequence of gene has gone little bit changed, because this chromosome has taken a part of this and this chromosome has taken a part of this. So obviously the sequence of genes which were there in our parents and now in us, Is it the same? No, there's a difference now. And because of these difference, we show variations. Because of these recombination, we show variation. Then what is variation? The small changes that we show in our characteristics as compared to our parents. The small changes that we show in ourselves, the small changes that we show in our characteristics as compared to our parents, because it may be believed something this way, that if we have developed from our parents, then we should be exactly identical to our parents in all characteristics. Meaning, if I talk about 50 characteristics, all should either match with my mother or should match with my father. But I get to see that no, almost about 40 characteristics does match, but there are some 10 characteristics which neither matches with my father nor with my mother. Then how do I explain those 10 characteristics that I have in me? These are variations. These are new characteristics that I have developed because of all these changes that has happened in the chromosome, and this has resulted in recombination and now development of new characteristics, which we will always have to define as variations. Okay, so these are variations that we basically understand. Now, one concept of dominance and recessiveness to understand, since we had already talked about the concept of homozygous and heterozygous, here we are talking about an example of inheritance where a characteristic that we talk about as tongue rolling. We talk about as tongue rolling. Now, tongue rolling, and there are many features to talk about like this. So this is one particular example. Tongue rolling, when we understand what is tongue rolling, a feature where certain individuals, they can actually roll their tongue absolutely, resulting in the formation of a tube. Can anybody do that? Both the sides of the tongue almost rolls up to form a tube. Can you? No? I think some of you can. If you try, you will be able to understand. So when we are talking about the rolling of the tongue, some of us we can, some of us I cannot. So when we are talking about rolling tongue, both the sides of the tongue results in the formation of a complete tube. That is called rolling of the tongue. Now, this particular feature, let us try understanding what are some of the alleles responsible. So, people with this particular feature, I would understand. Let us assume or let us consider capital R as a allele responsible for tongue rolling. Okay, responsible for tongue rolling. So now, when we are talking about the three types of genotype. Now, let me talk about three types of genotype. Here I am talking about individuals. If it is having a homozygous recessive genotype, then these are non-tongue rollers. They are non-tongue rollers. These are tongue rollers. These are tongue rollers. What about this? These are also tongue rollers. So that means when we talk about tongue rolling is regulated by a dominant allele. Tongue rolling is regulated by a dominant allele, and when we are talking about tongue rollers, individuals can have two different types of genotype. People who are tongue rollers can have two types of genotype. What are the genotype? They may be homozygous dominant, they may be heterozygous. And if they are homozygous, or if I talk about a person who is a non-tongue roller, then that person is always a homozygous condition. Now, this we can also represent in the form of a pedigree chart. Now, what is a pedigree chart? In case of a pedigree chart, we are actually showing generations with the help of some boxes and circles. Let us now do that. How do we show certain type of a condition? Here, certain rules that we have to follow. I talk about a box here, and then circles here, and then circles which are shaded. I'll just tell you the conditions, and after that I'll ask you to work out. You will try working out. Now, why did I draw this? Let me just show you all the four conditions. Now, all the 4 conditions that I'm talking about is, I talk about a box here. How do we represent generations? This is a box here. The box would generally represent a male who is not having the trait that we want to express, that we want to exhibit. So now I want to exhibit the trait who are non-tongue rollers in one particular generation. I want to see, or in 2-3 generations we can see, tire by tire we can see. So here, first of all, try understanding. I want to find out in 1 or 2 generations how many of them are non-tongue rollers. So people who are non-tongue rollers, or the trait that we want to show, should always be expressed in shaded form. Trait that we want to show in that entire pedigree chart should be shown in the shaded form. And if it is a circle, circle represents female, square or the cube represents male. If the male or if the cube is empty cube, we are talking about that male does not show that trait that we want to exhibit, that we want to show. So that means I have to talk about, now let me just talk about an example. Let us talk about an example to do. Here, with all this information, the empty square, the empty circle, the shaded square, what does they signify? The male not having the trait, the female not having the trait, female having the trait, male having the trait. All the 4 conditions. Okay. Now, say suppose I talk about a marriage between 2 individuals like this. What do you understand?
Then both of them are not having the same.
So I said we are trying to, any you can pick up. Now I'm talking about we try to show in this pedigree chart individuals who are non-tongue rollers. So now when I talk about a marriage between this, what do you understand? A female who is a tongue roller, a male who is a tongue roller, because we try to show non-tongue rolling, and they have 4 children, and out of these 4 children, now that I talk about, out of these 4 children, 1 is a non-tongue roller. Okay, I'll give you the condition, you will just work it out in your notebook. Take out. So this is a condition that I talk about, parents, these are the parents. And now here you will write down exactly the condition. I'm just writing it here. Female non-tongue roller. Female tongue roller. Male non-tongue, male tongue roller. So now you put the respective symbols. Put the respective symbols now here. According to this type of a pedigree chart where we are talking about 2 generations. These are simple questions sometimes you are asked in the paper. How can you interpret? How can you read? just correct this. It cannot be non-tongue roller. It will be tongue roller, the fourth one. Now this genotype is optional for you. That is not actually shown in your book. Genotype, if we select this type of genotype, then this is what we are supposed to do. You just write down the symbols. Now tell me, what should I write this symbol as?
Shaded.
Circle and shaded. This should be circle and shaded. What about this one? Hollow. What about this one? What about this? Hollow square. Understood? So what are we talking about is, so this we should draw here, here, here. So this is what we are talking about. But if the parents are like this, then what is the probability? Now here actually this is not a crossing. The crossings we'll do later. Now this is actually not a crossing. Here they will straight away give you that the parents are this and these are the children that they have, where you should only remember the symbols and put the correct symbols. I have just worked out to understand what can be their respective genotypes, but this is not a typical cross. Here only this condition will be given, this will be given, and or the symbols would be given and you will have to write this, or the phenotypes will be given and you have to put the symbol. Either of the type of questions you get. This is given, and this you have to do, or this is given, or this you have to interpret. So this is a typical question. Genotypes I have just written not to take it, not to get confused. This is not a genotype question. It's not a cross. So this is a basic concept of pedigree analysis which you are at times asked. Now we straight away enter into a concept of X-linked inheritance. Then after that we'll move on to the laws. We now talk about a concept of X-linked inheritance. What is meant by X-linked inheritance, since we had already talked about the concept of chromosomes, sex chromosomes. Now we try understanding that in the human population, there are certain traits which pass by the way of X chromosomes. In the human population, there are certain traits which would pass on from one generation to another by the way of X chromosomes. What are those traits called? Those are called X-linked traits, and since they pass on from one generation to another, we can talk about them as X-linked inheritance. Also, they are known as sex-linked inheritance. X-linked inheritance or sex-linked inheritance. What are some examples of X-linked inheritance? We can talk about examples of hemophilia. We can talk about examples of color blindness. These are examples of X-linked or sex-linked inheritance. What does that mean? Traits which pass on from one generation to another by the way of their X chromosomes. Now, one interesting concept here is that if I'm talking about the passing over of the characteristics from the parents to offsprings by the way of the X chromosome, always you will come across, if it is the X chromosome of the father, the X chromosome of the father, the son will not get. The son will not get the X chromosome from the father. The son will always get the X chromosome from the mother. We'll try understanding now. And again, if I'm talking about the X chromosome of the father, the daughters will always get the X chromosome from the father. So here the concept is, why talking about X-linked inheritance and also known as crisscross inheritance, because the X chromosome from the father passes to the daughter and then to the next generation grandson. The movement pattern is always that way. Sons, they never get their X chromosome from the father. Understood? So let us try working out one or two examples. First of all, let us start with the concept of hemophilia. Let us talk about a concept of hemophilia. Parents. The two parents that we understand, you are only to focus on the 23rd pair of chromosome. We are only talking about the 23rd pair. So how will we write? I talk about the female first. I talk about the female first. a pair of X chromosomes, a small cross. Then I talk about X and Y. I'm talking about the testes, I'm talking about the male, and I'm talking about the female parent, I'm talking about the male parent. And we can generally write a small circle here indicating that this particular X chromosome carries the recessive allele which is responsible for hemophilia. So there are different ways of writing. Either we write this or we can also write this way, where this small h is actually talking about the allele for hemophilia. Hemophilia, do you know what is hemophilia? Have you heard about hemophilia? What it is? Hemophilia is a
condition where there is a blood loss phase.
Like for example, if a person carrying hemophilia, if he got an injury, the blood will not stop and like it will not get clotted.
I talk about hemophilia, which is a genetic disorder. We are talking about X, so obviously understood. It's a genetic disorder that we are talking about. Now, in this genetic disorder, what is happening? This genetic disorder is caused by a recessive allele. Recessive allele. When I repeatedly say that this is caused by a recessive allele, what do you understand? A recessive allele will always get expressed only in homozygous condition. So try understanding this concept. When I'm saying that hemophilia is a recessive allele, that means in a male body, again, this zero that I'm talking about, the knots that I'm talking about, representing the recessive allele responsible for the disease. So a male may have a chromosomal complement like this. At the same time, a female, if it has a chromosomal complement something like this, try understanding this concept before we start with the cross. It is possible that males generally suffer from hemophilia. It is quite, I would understand 1 in 50 individuals do suffer from hemophilia. So, but in females, very rare, maybe 1 in 200 or 1 in 1000. So when we are talking about in human females suffering from hemophilia is almost next to rare. Because we have a pair of X chromosome. And if at all in one of the X chromosome this recessive allele is there, the dominant counterpart, the dominant allele will obviously be there in the other X chromosome. Yes. The dominant allele, so we generally represent the dominant allele by plus. So this dominant allele which is there in the other X chromosome will inhibit this allele from expressing. Even if anybody of us has this type of a combination, maybe we have got this allele. Will this allele ever express? Generally not, because the other dominant allele will always suppress it. What about the story in case of males? In case of males, one is, if this is the X chromosome, this is the Y chromosome, the two chromosomes X and Y, they are of differential length. The Y chromosome is the smallest chromosome, almost half of the length of the X. So now, if I am talking about that the allele for hemophilia is here, is there any chance that the dominant allele is there in the Y? Generally not, absolutely not. The dominant allele for quite a number of alleles which are there in the X chromosome, the Y chromosome does not have it at all. Because number one, the Y chromosome is very short and many alleles which are there in the X does not have its counterpart in the Y. So because of this, now this recessive allele can easily express, because this recessive allele has no other allele to suppress itself. So it is expressing. So this is one big reason why in the male it is expressing and in the female it is not. And such type of a condition is also known as a hemizygous condition. Hemizygous, where one allele is here, the other allele is absent altogether. Am I clear? So now, based on this, we come across, if I have to talk about color blindness, also the same concept. If I talk about color blindness, also the same concept. And now to understand hemophilia a little bit, males who suffer from hemophilia, they may profusely bleed even from a smallest cut. Quite often, whenever we are brushing our teeth, it bleeds, but it bleeds for a couple of seconds, and after that, automatically it stops. But for people suffering from hemophilia, that may be a good reason for hospitalization, and if not taken care at the right time, people do collapse also for this. Profuse bleeding, because they lack a particular type of a protein factor, which brings about the process of clotting very fast. So because of that, they have to be immediately given some coagulants, injections and etc. has to be given immediately. If that is not done, many individuals, they suffer from acute conditions also. So that comes to an ICU condition and a proper medical condition it comes to. So this is...
Albinism also be part of extinct inheritance.
What is this?
Albinism.
Albinism is different. Albinism is not because of this X-linked inheritance. Albinism is just somatic inheritance when we talk about, where it is because of certain alleles where the pigmentation in the hair and the skin and the eyes absolutely absent. That is a different, of course it is also a genetic disorder, but not a type of genetic disorder like this. This is exceptional. So now we go with the cross. How should we do? These are the parents. When we talk about the pair of chromosomes, we should be writing this way: a pair of X, a pair of Y. Now the X, Y, I'm sorry. Now we talk about the gametes. Let us try understanding how to write down the gametes. How do we write down the gametes? This female parent will produce how many type of gametes? This female parent will produce how many types of gametes? How many types? How many types of gametes this female? One type. Repeatedly I told you, females have XX, the same pair of chromosome. So even if the number is 4, the type is 1. Of course we are talking about meiosis and 4 gametes. But now I am asking you only one type. So how many types of chromosomes? I would write this is 1 and this is another. We generally can circle and write because gametes can be represented this way also. We now talk about this. How many types of gametes here? How many types of gametes here? This is one type and this is another. So 2 types of gametes for the male. Then now we are talking about what would be the F1 generation or the offsprings now. In case of human, we talk about offsprings. What is the parental combination we took? The parental combination we took is a normal female. So what are we supposed to write actually is, we would write this is a normal female. Try understanding. I will give you one more question to solve. Normal male. And what about this one? This is actually a hemophilic male. It is a hemophilic male. Why do we say it is a hemophilic male? Because the allele is there. Okay. Now we talk about the crisscross method and the Punnett square. Both the methods we are going to do. Let us talk about the crisscross method first. What am I doing? I just show you for understanding. 1, 2, 3, 4. I was numbering them. So what am I doing here? Crisscross method would mean when 1 and 3 undergo the process of fertilization. Then what would be the combination? 1 and 3, X and X. It will be something like this. Clear? I talk about now 1 and 4. I'll finish with 1 first. 1 and 4. Do I get something like this?
Yes.
Correct, no? Now I take 2 and 3. Can I show fertilization between 1 and 2? Why not?
Because they were same.
Gametes of the same parent. So always remember, I am taking 1 and 3, 1 and 4. Now I'll take 2 and 3, 2 and 4. So I take 2 and 3. What will be the combination? X and 0. I take 2 and 4. What will it be? X and Y. Now we are supposed to write down the phenotype. What should be the phenotype of this?
It will be an affect.
It will be considered as a carrier. The female is having the allele, but will the female suffer? The female will not suffer. So we will write this is a carrier female. This is a carrier female. What about this one? Normal male. This is a normal male child. What about this one? And again, a carrier female, a carrier female. What about this one? A normal female. Take a note here, I have been talking about this X chromosome. I had been talking about this X chromosome. So this X chromosome, did the sons got it or the daughters got it? The daughters got it. Check here. This X chromosome the daughters got. This X chromosome the daughters got. And again, if I follow the next generation where I would say that this carrier female marries a normal male. This carrier female marries a normal male, then you will come across that the sons are getting this X chromosome. So that's the reason it is known as crisscross. One generation suffering, next generation skip. Next generation nobody will suffer. But in the next generation again, there will be sufferers. So it is always skipping one generation and then sufferers will be there. So are you clear with this one? Let me just show you the Punnett square method, then I will also ask you to continue from here. Now if it is the Punnett square method, then what am I supposed to do? If I have to show the Punnett square method, then what am I supposed to do? From here till here exactly same. Here only there is a difference. So what am I supposed to do is, I will make a box like this. This is known as the Punnett square. And how many columns and how many rows? This is what my box is. So here I am going to write down the male, female gametes, and this side I may write down the male gametes. So what are the gametes for the females? Tell me what are the gametes for the females? 1 and 2. No, these are the two gametes. So 1 and 2 I write down. What are the gametes for the male? This is 1 and this is another. So I have written down the male and the female gametes. Now what did I do? Fertilization. 1, 2, 3, 4. No. So this fertilized with this one, this fertilized with this one. So I'll have to write it this way and this way. Then this with this one, this with this one. So I write this here and this here, right? So now exactly the same way I'll have to write down the phenotype and phenotype here done. Now you get questions like this, that what is now under such type of a condition where the normal female is crossed with a hemophilic male, or a hemophilic male marries a normal female. What is the percentage of sufferers? No sufferers. What is the percentage of carriers?
2 out of 2.
How many offsprings are carriers? 50%. 50% of the offsprings are carriers, no? And if I ask you what is the percentage or how many female offsprings are carriers, all are carriers. All are carriers. What is the percentage? Female, then all. And what is the percentage of normal offsprings? 50%. 50%. Got it? Now you continue. Take the question.
And will they ask the ratio also?
Generally, percentage they ask. What is the percentage of female offsprings? What is the percentage of offsprings? So if it is offspring, then you have to take account of 4. If it is female offspring, you have to take account of 50%. Out of 50%, how much? How many? Okay, now let us continue from here. Now if you have understood this, let us continue from here. Take the question. What will be the percentage of, take the question, you write down the question. What will be the percentage of offsprings? What will be the percentage of offspring suffering from hemophilia? What will be the percentage of offspring suffering from hemophilia if? What will be the percentage of offspring suffering from hemophilia if a carrier female? If a carrier female marries a normal male. If a carrier female marries a normal male. The cross is in front of you. You will do it exactly step by step that I have shown. But you'll have to remember, carrier female and a normal male you have to take now. Understood, no? Now I showed a cross between normal male and hemophilic, normal female, hemophilic male. But now you have to take a carrier female and normal male. Do it. And you will show by the Punnett square method. This method. This is most commonly asked in the paper. Start very neatly. You will do exactly the steps that I showed you. Very neat. Write down the phenotypes, genotypes nicely. Make the Punnett square neatly. And why do you start from here straight away? How did I show you? Parents, gametes, offsprings. Exactly this way you have to write. Don't start from the Punnett square straight away. You also. You have to start exactly from the steps that I'm showing. Yes. You'll have to show exactly what is my question. I gave you a question. What is the percentage proportion of offspring suffering from hemophilia when a carrier female marries a normal male? So now you'll have to take here carrier female, normal male, and exactly what I have done. This is what you have to show. It is a 2 to 3 mark question. If you only do the box, you will not get more than 1/2. All the steps write down properly. What are you calculating there?
I'm writing about this.
Are you showing all this? You'll have to show me, huh?
I'm writing down.
Wait. Exactly step by step. See that you don't get confused. If there's any confusion, you will ask. Again, I repeat, exactly what I have written, this is what you will write down. Let me put it clearly here. So what do I take? I take this way. Yes or no? I take the female this way. So now this becomes a carrier female. So this becomes a carrier female now. And what am I supposed to take? This is a normal male, I told you. So now you go about. Are you understanding? Any doubt?
What was the mention of offspring circle?
Exactly same thing. Now write down the gametes. What should be the gametes, right? After you are done with the gametes, now check offsprings, what it should be. Not following? You have a confusion. I can understand from your face. What is the confusion? Tell me. You will first write down the genotype. Is this clear? This is done. So genotype once I have written, I have to write down the gametes. What will I write down? Gametes means what? This pair of chromosomes will just get split. So this goes in the formation of one gamete. This goes in the formation of another gamete. So this is for the female. Same way I write for the male.
And then how to represent the offspring? So do I have to also make lines as well?
I told you can do by this method. Punnett square you do. Sit down, I'll come to you. So now this Punnett square you'll have to do. That means what? I have reached till this part. Now this I will not do. This crisscross method I will not do. Straight away from here I told you, you will go to this box. Make this box. Write down the male gametes, the female gametes. And then keep writing down the combinations. Anybody has a doubt with this? Girls? Any doubt with this?
I did the both.
You did both. Very good. See? This side, boys, anything doubtful? This is one of the simplest cross that we are doing. Now that we talk about Mendel's crosses, it's going to be a little bit even more confused. तो अगर ये नहीं समझ में आ रहा है, please ask. What are your names? Tell me.
Yajat.
Yajat. Pragya. There. You all play some sport?
I went to cycling.
Huh?
I went to cycling.
Huh? Cycling. Pragyan and
Cricket
sometimes. Our green khelna hai. See, look at your health. You'll not be able to lift up only books in the 5th, 11th standard. Huh? Your parents have to carry books for you. And even that one, see. Strong. ICSE 10th, 11th, 12th, if you are planning to go with ICSE 11th, 12th, not so easy. Done?
Yes, sir.
Pragyan, are you done with this? Tell me, what should I write? What should I write here? Tell me. Gametes I want to write. Sorry. Gametes I want to write. Female gametes. What should I write?
I wrote female gametes.
Anyways, male gametes, what should I write? Normal male. Normal male. Okay. X and Y. Female gamete, I said that's a carrier female. So what should I write?
X and
X0
X0.
and X, no? Okay. Then now what should I write in the first box? Tell me. What should I write in the first box?
X and X0.
X and X0. Okay. Then
XX.
here?
Yes.
Okay. XX. Tell me what should I write here?
X0 and Y.
X0 and Y. And here?
XY.
XY. Here, what is the phenotype of this one? Tell me.
I'm XX0.
Phenotype.
Carrier.
Carrier. This is a carrier. What about this?
Normal.
Normal female. What about this?
Carrier.
Sufferer. This is hemophilic, no?
Oh.
This one is hemophilic, no? Male, no? And the recessive allele is there, so it is hemophilic. So either sufferer or hemophilic you are supposed to write. What about this?
Normal.
Normal male. My question was, what is the percentage proportion of offspring suffering from hemophilia? What is the answer? Percentage proportion of, what is the percent? What percent of offspring suffer from hemophilia? What percent of offspring are suffering from? You have to take a 4. Out of these 4, you have to consider 100. And then what percent of offspring suffering from hemophilia? कहाँ पे 1% का
only the lady.
हाँ तो 1 out of 4 that becomes 25%. 25% suffering from hemophilia. What percent of male offspring suffering from hemophilia? What percentage of male offspring suffering from hemophilia? 50%. Are you clear with the different types of questions? If it is male only asked, then 50% suffering from hemophilia. What percent of offspring suffering? 25%. These are the two type of questions you get in the paper. Clear, no? Clear? So if I go from the first cross, what did I see? That in the first cross, the male was hemophilic, and the sons, no one was hemophilic, the daughter was carrier. And if the carrier daughter marries a normal male, then the son again is hemophilic. So that's why we talk about this pattern of inheritance as crisscross inheritance. Now we talk about the concept of Mendelian inheritance, the concept of Mendel's laws. What are we supposed to understand? Okay, before that, can you complete this progress check there? Question number 3, page 31, progress check. Question number 3 and 4. Question number 3 and 4. Will not take more than 2 minutes. Done? What is answer? Third. Question number 3. What is answer?
27.
What is the question?
Mention the number of paired homozygous chromosomes in human female.
Mention the number of paired
Paired
homozygous chromosomes in human
Female. The first and second is human being.
paired.
Subquotients.
Mention the number of paired homozygous chromosomes. Paired homozygous chromosomes in human female. Paired homozygous chromosomes in human female is
20.
paired homozygous. When we talk about, what do you understand? Homozygous, what do you understand? It is not homologous. It is homozygous. Homozygous chromosomes that we are talking about. Mention the number of paired homozygous chromosomes in human female. In male it will be heterozygous, it will not be homozygous. No? In male it will be heterozygous, no homozygous. So paired homozygous chromosomes. Paired homozygous chromosomes. Homozygous is only we have to talk about the concept of X and Y. Not autosomes. Autosomes, when we talk about all the autosomes, then we have to consider them as homologous. These are the homologous chromosomes that we are talking about. So if it is homozygous, remember I already told you the concept of homozygous and heterozygous and hemizygous. Three concepts are there. So if I have to talk about the homozygous, not to understand autosomes here. Autosomes we will not write here because when I talk about autosome, that becomes the concept of homologous. Homozygous in case of human, female, one pair, and in case of male, zero. Homozygous in case of female, males doesn't have any homozygous pair. They have homologous.
Generally, like the sex chromosome.
Sex chromosome 23rd pair only.
Okay.
In case of female, it will be one pair. That is your answer. In case of female, it will be one pair, and in case of males, no, they don't have zero. In males, there is no homozygous pair at all. It is hemizygous rather. It is called hemizygous, where one X long and Y is very short. Irregular chromosome it is, X and Y. Okay. Come to the next question.
Can there be a heterozygous recessive? Explain.
Can there be heterozygous recessive? I explained you this also. This I basically explained you the other day. Can there be heterozygous recessive? I talk about a pair of chromosome and I am talking about A and A, heterozygous. I talk about A and A, homozygous. I talk about A and A, homozygous. How do I write heterozygous recessive?
There couldn't be a heterozygous recessive.
Nothing possible.
As in that case, only the dominant allele always should.
When I talk about heterozygous, heterozygous always means heterozygous dominant. When I talk about heterozygous meaning, it is the dominant which is expressing. So when I'm talking about heterozygous, it has to be one dominant, one recessive. And there is no way of expressing two recessive and showing a heterozygous. Not possible. Understood, no? We can never express any heterozygous but a recessive condition. The moment you talk about heterozygous, it always means one recessive, one dominant. Yes or no? So that's why we cannot write any, there is no heterozygous. Write down what is the explanation that you should write. What is the question there?
Can there be a heterozygous recessive? Explain.
Write down. No, comma. What is the explanation? No, comma. Heterozygous combination would always have. Heterozygous combination would always have. Heterozygous combination would always have one dominant allele and one recessive. One dominant and one recessive. Heterozygous combination would always have one dominant, one recessive. Now let us start with Mendel's laws of inheritance. What are we supposed to study here? We will be studying three laws here. One is the law of dominance. We are studying the law of dominance. What would be the second law? The law of segregation. The second law is known as the law of segregation, also known as the law of purity of gametes. The law of purity of gametes. And the third law is known as the law of independent assortment. The third law is known as law of independent assortment. Let us start with the laws one after the other. Try to understand the concept. I have already discussed all the important definitions and concepts and process, also sex-linked inheritance. So understanding this should not be difficult at all, provided we have understood the previous concepts. Now, if I have to a little bit give you an introduction about the concepts of inheritance, here Mendel had been experimenting on certain plants. Okay, so if I have to talk about who was Mendel, Gregor Johann Mendel, that we understand, a very famous botanist actually. Gregor Johann Mendel, that's his full name, and he had been working in his monastery, and in his monastery he had been observing a variety of plants, experimented quite a number of them. But one such type of plant attracted his attention was the pea plants, the garden pea. You are asked in the paper, give the or write down the common name. Common name, when you are asked, you are supposed to write down the garden pea. And biological name, when you are asked, you are supposed to write down Physarum sativa. That is the biological name, and just underline as per the rule of nomenclature. So these are the two common questions you get in the paper, 1 mark. So give the common name, garden pea, and then the scientific name, Physarum sativa. Now, what was the reason that these plants attracted his attention? Because these plants exhibited some contrasting traits. Now you know what are traits. So these plants exhibited some contrasting traits. And contrasting traits in the sense, sometimes some plants were tall and some plants were quite short, dwarf. Some plants gave rise to some white flowers, sometimes plants gave rise to purple flowers. Sometimes the seeds were green, sometimes the seeds were yellow. So like this, there were some 7 pairs of contrasting traits that he observed in these type of plants. Otherwise, when we talk about most of the plants, in most of the plants you will come across the flower would be more or less of the same color always. The seed would be more or less of the same shape, same color always. But in this type of a plant, he saw sometimes the shape of the color and the seed and the position of the flowers and the height of the plant, not the same every time. They're quite contrasting. So he started self-pollinating them. What is the meaning of self-pollination? Pollination between the flowers of the same plant. So he started self-pollinating, and every time he would observe new characteristics. What were those 7 pairs of characteristics? Let me read out, and you will please take a note of this. What were the 7 pairs of contrasting traits? What were the 7 pairs of contrasting traits? The 7 pairs of contrasting traits, check. I talk about the character. What is the character? Height. If I talk about the height as the character, what were the traits? Tallness and dwarfness. Talk about the color of the pod. Color of the pod. What were the colors?
Green
and yellow. So the first line of traits are the dominant traits. So take a note. So green is dominant, yellow is recessive. Shape of the pod, inflated and constricted. Inflated means fleshy. No? So commonly the pea that we get to see has got a fleshy leaf. No? And constricted, sometimes we get to see some dried, papery, but the seeds inside are fresh. So that is about the color of the pod. Shape of the pod, inflated, constricted, I told you. Color of the seed, yellow and green. Shape of the seed, round and wrinkled. Position of the flowers, axial and terminal. Color of the flower, purple and white. So these 7 pairs of contrasting traits of every time he used to observe, and that's why he started crossing these plants, and he came up with certain conclusions, certain ratios of expression of the traits. That is what he put forward as these 3 distinct laws. So let us talk about the first one, that is the law of dominance. Now, when I talk about the concept of dominance, you already know what is the meaning of a dominant allele. What was the basic experiment that Mendel did was, Mendel, I'm not writing down the cross yet. Mendel took some tall plants and he crossed them with some dwarf plants. In the first generation, he found all plants were tall. That was his basic experiment. So he took some tall plants, crossed them with dwarf, and in the first generation, he saw all plants were tall, not even a single plant dwarf. So initially, he thought there must be some mistake. He does it again and again and again. Then he changes the traits, and then he does it purple and white flower. All plants with purple flowers he gets to see. Round seeds with wrinkled seeds. All plants with round seeds he gets to see. So he every time saw that when 2 plants with 2 contrasting traits were selected, only one trait was getting expressed and the other trait was always remaining suppressed. But he was very much sure that this trait must be there in this generation, in these plants. So what he did later, he took plants of the same generation and again crossed them. Then in the next generation, he saw there were a huge number of plants which showed one trait, but now there were some plants which were showing the other trait also. So that means this trait entered into this generation but did not somehow express, and now expressed in the next generation. So this was some of his concepts that he understood and which happened to be quite applicable even when we talk about human population. So now if I talk about the mother has one particular trait, the father has one particular trait, we get to show or express only one trait. Yes, this is applicable in human populations also. And yes, certain traits does not get expressed but hidden here, but may get expressed in the next generation. So this is what we are talking about, the law of dominance as well as the law of segregation in this concept. After the break, when I come, we'll start writing down how this process should be written exactly. Okay, we'll continue. Take a break, we'll continue.
ये सभी आता है
Fine.